Okay, let's start. My name is Chen Zhao. I'm a research scientist from NVIDIA, and I will host today's MICCAI Industry Talk. I feel honored to introduce our speaker today, Dr. Alvin Chen from Philips. Dr. Alvin Chen is a senior scientist and group lead at Philips, where he specializes in the development of AI-powered ultrasound imaging and diagnostic technologies to improve healthcare access and emergency response, particularly in point-of-care settings. His team focuses on developing and applying machine learning techniques and best practices to address challenges in medical data, annotation, and clinical validation of these technologies. Dr. Chen has co-authored over 30 peer-reviewed publications and more than 50 patents and patent filings. He earned his PhD in biomedical engineering from Rutgers University. Let's welcome Dr. Chen. Yeah. Hi, Alvin.
Okay, I think I'm not sure if it was on my side or the
Sure, please.
audio broke up a little bit, but yeah, thanks Khan for the introduction and thanks everyone for joining. I know folks are joining from different time zones, so I really appreciate your time. So for this talk, I wanted to talk about some of the experiences that we've had, our team has had recently in our work in developing AI models for healthcare applications. And in particular, what we've found is that there's some real world challenges in translating AI from the benchtop to the clinic. And so I wanted to address some of these and share some of our learnings, particularly related to data annotation, models, and validation and real world testing. And before I start, I just wanted to highlight that this is work being done by a large team, and so I'm only speaking on behalf of the team. And very quickly, just an acknowledgement of some of our funding sources from the Department of Health and Human Services, as well as from BARDA. So I think all of us are aware that AI has really blossomed in recent years, and one indication of this is the explosion of scientific publications related to AI in healthcare. We can see from this graph that the increase really is exponential with tremendous uptake in the past several years. What's interesting is, at least from a US-centered perspective, the translation of this incredible work into FDA-approved AI medical products still lags behind. So we can see that there is a translation gap that still remains. And the number of publications still continues to greatly outpace the number of approved devices. So the question is why is this? And is there something that we should think about or consider to try to bring more of this work into the clinic? And this certainly has been a topic of a lot of discussion and thought. There's been a number of publications in the last just two or 3 years exactly talking about this question. One in particular, the paper by Raj Prakar from 2022, I thought had a really nice quote where they highlight the fact that deploying medical AI systems in routine clinical care is faced with complex ethical, technical, and human-centered challenges. And I just grabbed one of the figures from the paper that proposes some different opportunities for addressing these challenges related to data, AI, and so on. And so this is the theme of the talk that I wanted to go through, and I'm going to try to break down the challenges that we faced into these 4 aspects. So there's challenges related to data quality and acquiring good representative clinical data. The annotation aspect, which is a huge part of the developmental effort. The model itself, and then perhaps most importantly, how to actually demonstrate that AI has real-world efficacy and improves human health. just as for some context for this work, I'm going to present most of the discussions in the context of one case study. This is recent work that our group has been doing related to detection and diagnosis of infectious lung diseases. So, very briefly, some background. Infectious diseases are one of the leading causes of illness and death worldwide, both in adults and children. Hundreds of millions of incidences each year, millions of deaths each year. Common causes include bacterial infections, virus. We all remember just not too long ago the COVID-19 pandemic, which is an example of an infectious lung disease that obviously had a huge global impact. And so the ability to detect and diagnose lung infections can be life-saving at times, can lead to tremendous improvement in patient outcomes. In terms of how diagnoses typically are done, they're typically done using imaging for viruses and bacteria. You can also have lab tests. Our focus is on medical imaging, and in particular, our group works closely with ultrasound. And we are interested in this modality because of its suitability for deployment in point-of-care settings at the patient bedside. Also, its suitability for resource-limited settings and developing areas. And so the talk here will be centered on our work in ultrasound, but I think a lot of the core challenges and experiences also apply to other applications and other imaging modalities. So what are we actually trying to do? So the ultimate goal is to develop computer-assisted detection and diagnoses of pathologies that are linked to lung infection and infectious disease. And what we want to do is that it's not a competition clinically between AI and clinical experts, but rather to develop a system where the two elements can work together, and that can lead to improved decision making, diagnostic accuracy, accessibility, and so on. And so the overall kind of approach is you have an ultrasound imaging system. This could be a portable system, for example, especially if we want to deploy it in point-of-care settings or in sort of resource-limited areas. And ultrasound is a video-based modality. So you typically acquire video loops that can have hundreds of frames in them. And a clinician will analyze that loop, look through, kind of scan through each of the frames, and similarly, as you would for other modalities, determine the presence or absence of pathology and use that information to provide a clinical diagnosis and develop a treatment plan. And so, of course, the images, these video images are provided to AI models, and the AI models provide computer-aided support. And so one approach to doing this is to have the AI run on each frame of the video and to output both a output at the frame level. So for each frame of the video, it lets the user know the presence and location of pathology. Also, the video as a whole, you can determine kind of based on the sum of all of the frames, the presence or absence of pathology, their location and severity, and so on. And in the case of this particular clinical area, there's actually a number of different pathological features that are observed under ultrasound, which clinically lead to differential diagnoses of a range of different conditions. And so it's really this team between the AI and the human together that forms the final diagnosis. One of the big learnings that we've had is that it's probably not the most prudent thing to try to have the AI do everything right now. And it's that their combined accuracy is expected to be higher than AI alone. So let's just go through each of these components piece by piece. The first is challenges related to data. And a couple of the questions that we face are what actually defines a good clinical data set? And part of that is ensuring that the data is diverse. It's important, very important that there's inclusion of less represented groups. And then another question is how to partition data for the development stage, and then separately the actual clinical validation step. So what defines a good clinical data set? The FDA and other regulatory bodies, including the Canadian regulatory body and the MHRA in the UK, actually have recently come out with guidelines for good machine learning practice. And a big part of those guidelines is about the data. And they've highlighted two uh, criteria for a good clinical data set. The first is that it must be representative. So I'll briefly read the quote, but the clinical study participants and the data set must be representative of the intended patient population. So that includes demographic information, clinical diagnoses, other measurements, and these need to be represented both in the training set as well as in the test set so that you can maximize generalizability. And obviously, the training set and the test set need to be independent. I think we, as AI practitioners, are all aware of this, but the degree to which they must be independent, at least for us, was a learning. So not only does obviously an image used in training can't be used in testing, but even beyond that, there needs to be ideally separation on the subject level and even on site and geographic levels. So we don't want to have results biased on particular sites or particular geographies. So you want a distribution, and you also want to keep the clinical sites and the data sources used for training separate from those used for validation. So I will go into some of these details in a bit. And so what we've done in trying to collect data that meets these guidelines is a structured prospective data collection effort. Over the past few years, the group has set up enrollments across the US. The work up to this point is focused on the US. The eventual goal would be to also demonstrate efficacy outside the US, but for the purposes of this talk, I'll focus here in the States. And so in total, we've acquired data from over 1,000 patients across 16 sites. You can see the geographical distribution. We tried to make effort to cover all geographical areas in the country to the extent possible. But not only geography, we also wanted to make sure that we're collecting data from all relevant hospital settings. So we know that patients with bacterial pneumonia, COVID, et cetera, they can present to the emergency department. They can also be admitted to the hospital. And even in severe cases, they may be admitted to the intensive care unit. And so the pathology, the disease, the patient, they're all a little bit different depending on the setting. And so we want to make sure that all possible settings in which the AI is intended to be used are represented in the data. And then one very interesting component of or element of this particular project is data drift due to the COVID-19 pandemic. So one finding was that image appearances and diseases actually changed before, during, and after COVID-19. And so an additional dimension was to ensure that there was diversity over time so that we had data representing broadly lung infections, but not biased one way or the other due to COVID. And just here's some detailed information showing that we try to observe and ensure diversity also across demographics. And we try to have a representation of different medical histories, different clinical diagnoses. So all of these factors we've learned are important to account for in the data collection, again, to ensure representation. Now, one question that might ask is why the need to collect prospective data in the first place, right? So depending on the modality, there may be publicly available data, retrospective data, and so on. What we found is that prospective data is pretty important, particularly in ultrasound where public data are more scarce. But even when we have retrospective and public data available, prospective data allows us to standardize and structure the data. We can ensure that there's a standardized imaging protocol that's comprehensive, that doesn't miss zones or areas that should be scanned, that the data are collected by properly trained professionals. And in addition to the ultrasound that we're working with directly, we also collect structured reference standards. So these could include other modalities such as X-ray and CT that may be acquired as part of the standard of care exam for patients that are appearing in the hospital and enrolled in our study, and even non-imaging data such as lab test results, clinical data, vital signs, etc. All of these are relevant when we want to establish a reference standard and establish a diagnostic ground truth. The other advantages very quickly are we can have more direct control over patient inclusion and exclusion criteria. If needed, we can collect serial exams, which may not be available in the retrospective data sets. We also want to make sure we're representing a wide range of imaging hardware. So in our case, different ultrasound systems, different transducer types. Ultrasound in particular is interesting because there's a whole array of equipment that could be used, and we need to ensure that they're all sort of represented properly in the data set. And finally, it's also possible to obviously combine the prospective data with prior data, and that only further maximizes diversity. How did we set up data for development versus for validation? So this is a very important step and ideally is defined early on. So in our case, we collected data from about 30,000 videos, about 1,000 videos.
Uh, Sorry, there are some technical issue here. We have told the speaker, and the speaker will rejoin soon. I saw some audience put questions in the QA box, and thank you for doing that. So for everyone, when you have any questions during the talk, welcome to type your question in the Q&A box. So after the talk, the speaker will answer the question one by one. Thank you. Yes, the recorded session. If we got permission from the speaker, then yes, the recorded session will be available. Thank you. And in the MICCAI Society email, there is a link that hosts all the previous talks. To ask a question from Rila, if the talk stopped, just log in. So it was paused due to technical issue. Now the speaker Alvin is back. Hi Alvin, we cannot hear you.
You hear me now?
Yes, we can hear you now.
Okay. Yeah, my apologies. I think it's the Wi-Fi on my side. I'll reshare the screen and keep going.
Great, thanks.
Okay. Hopefully the screen share is working.
Yeah, this is working, and we
Okay.
can hear you. It works great.
Okay, great. So I'm not sure where we got disconnected, but we were talking about our work in the steps we took to separate out development and validation data and perform cross-validation in the development portion. And then finally, once we actually develop and freeze the models, we then test the performance of the models on the left-out clinical validation data set. And the validation results are what are ultimately reported for regulatory review. Okay, so those were the some of the observations and challenges related to data. The other really significant aspect of development is annotation. I think in particular in healthcare, what we've learned is that annotation quality is crucial, and really the models are defined by their labels. And so I wanted to kind of use as an example one publication by Raj et al. Also from 2022. This was a really interesting study where they looked at laparoscopic video data, so not ultrasound, but still a video modality from a large public data set. And they had a huge effort to annotate the data by both medical professionals as well as, interestingly, Amazon crowd workers. And they wanted to see the effect of the different types of annotation on the ability, on the agreement among each other and the ability to train AI using that. And a couple of findings that I found really fascinating were, firstly, that how you actually set up labeling instructions is tremendously important. So they found significant impact of labeling instructions on the variability of annotators with each other. One example about how they propose to have sort of useful labeling instructions is including images. So just something as simple as including example images as part of the instructions led to a significant improvement in annotator agreement compared to just text-based annotation instructions. Also, how you describe the annotations also made a big difference. And then the last kind of observation, which interestingly we've also, I think resonates with us, is the importance of the expertise of the annotators. And so the graph on the bottom shows professional medical annotators that they recruited from a couple of different professional annotation third-party services. And they compared that to the annotator agreement and error rate of Amazon workers, and there was a significant difference. And maybe not surprisingly, but the medical annotators performed better than the crowd workers. And the reason I bring up this work is because I think we experience a lot of similar observations in our own work. One interesting thing about ultrasound and lung ultrasound in particular is that there's a relative lack of established clinical society guidelines for how to rate these pathologies in ultrasound. And so one of the very first parts of this work was to actually develop a standardized grading scale. And so this was this kind of clinical groundwork that was done even before AI training started. And so, yeah, we developed a grading scale. I won't go into the details of the scale, but it's briefly shown on the right-hand side, where we are able to standardize the assessment of severity of a number of different lung ultrasound features. And as part of this work, we also looked at how, similarly to the Raj paper, inter-rater agreement among expert annotators. One of the interesting findings was that even among kind of world experts, um, iterative calibration and training and standardization of the labeling instructions and how each individual interprets labeling instructions was important. So this graph here shows that we uh, conducted a series of calibration sessions with this team of annotators, and that their agreement with each other actually improved over time with each session. So these are physicians with the tremendous experience in ultrasound, but just agreement in the particulars of the definitions of the annotations still were greatly impacted by how much training and calibration was done. We see a significant improvement, after several rounds of calibration compared to before, which was surprising, but also really important to know. And what this kind of taught us is that before we start annotating data on a large scale, we have to make sure that we properly calibrate, properly standardize, and train the annotation team. So standardization, reader calibration are essential for minimizing disagreements and ensuring reliable labels. The next question then is, so if you have multiple independent annotations, let's say you have 10 annotators, each annotating a set of images or videos, how to determine a final label for training, right? So here we looked at the consistency among these experts and how well they agree with one another, kind of on a paired individual basis. That's the first row. We also look at how well each annotator agreed with the mean of the remaining annotators. So we take each annotator, then we take all of the remaining annotators. In this case, there's 9 remaining annotators each time. We grab a kind of consensus or majority or mean vote from that remaining panel. And then we repeat this. We kind of do a round robin and repeat this across all the annotators and evaluate their agreement that way. And then finally, how about agreement between means of two groups of annotators? So the reason this is relevant is because what we found is that agreement between majority or mean-based annotations are more reliable than any individual. And this is shown on the right half of the table. So the left half of the table was the previous plot where we had multiple rounds of calibration. And then after the final calibration session, we looked at these different methods of measuring inter-rater agreement. And what we found is that consensus labels derived from multiple experts, for example, based on an average or based on a majority vote, exhibit higher reliability than labels from individual annotators. And this is related to AI because if we then can train the AI on these consensus or majority-based labels, those likely or potentially are of higher reliability than any individual. And that also points to one potential utility or advantage of AI is that AI can learn from a consensus, whereas a human alone is mostly reliant on their own individual interpretations and expertise. So to bring this kind of portion full circle, what we did was then with these findings set up a scaled up annotation workflow that involves multiple experts. So in total, we had 20 annotators. These are typically emergency and critical care physicians. We involve up to 5 annotators per video so that we can derive a majority from their independent annotations. And yeah, ground truth labels based on majority vote or mean. And then after we have a ground truth label, then we then proceed to downstream steps such as the annotation of individual frames, quality checks, reviews. And at the very end, there's always a physician sign off. They basically put a check mark and say that this video has been properly annotated and reviewed. And using this workflow of multiple experts, we annotated a total of around 15,000 videos. Again, each video is a series of frames. So this added up to over 1 million image frames in total from 400 something patients. And this was the annotated data set for model development. And this was work done by a large team over the course of several years. Okay, I'll move on to some learnings about the actual design of the model itself. And I think there's 3 points that I wanted to highlight. First is tailoring models to work with limited label data. So in our particular case, over time, we were able to build up a large label data set for supervised learning. But at the beginning, we didn't have that. And in many applications and many projects, a large data set may not always be available for training. So what happens when the data set is small or the labeled portion of the data set is small? The other thing is data imbalance. And then finally, how to actually compare the AI with human experts. So in terms of data scarcity, so ultrasound is challenging for one reason in particular is that frame-by-frame annotation of video data is extremely time-consuming. To go, label each frame with multiple features, I think we estimate on average that a single video loop takes about an hour to frame annotate. And so for 15,000 video loops, that's roughly 15,000 hours of work. And if this is multiplied by multiple annotators, that only grows even further. Comparatively, just annotating on the video level is much more efficient. So that can be done on the order of a minute. And then, of course, unlabeled data doesn't require annotation time. So typical supervised learning, of course, would focus on the fully annotated data set. You train, for example, a pathology detector, a segmentation model, et cetera, on the fully labeled data. But what we wanted to, and many others, of course, have looked into are ways to leverage either partially labeled data or unlabeled data, for example, using self and semi-supervised techniques. And I think what we found is that those really represent powerful opportunities for maximizing usage of data, particularly when the fully labeled portion is a small percentage. And so I'll talk about some recent work in this direction. The first is working with unlabeled data. So early on in the project, when we had much more unlabeled data than labeled data, we wanted to see, can we still leverage the unlabeled data to at least learn something? And so some of you might be familiar with these contrastive methods where we can pre-train a model using paired unlabeled data through different augmentations and learn kind of pre-learn feature embeddings that are relevant to the task and agnostic to the augmentations. So this is a common example where you augment an image 2 different ways, and then you have a shared network which learns features, and then you try to maximize the similarity of those features. So if it's the same image with different augmentations, you try to maximize their similarity and bring them closer together in feature space, whereas different images or negative counterexamples would be farther apart in feature space. This is sort of standard contrastive learning. And what we did was we applied some of these approaches to pre-train the models and then use those pre-trained models for downstream tasks rather than training the supervised downstream models from scratch. And so, for example, downstream tasks in our case could include pathology localization, so locating the area where pathology exists in each frame, as well as the video level classification. And what we did was, this was earlier on, we pre-trained models on a large retrospective data set that had been available at the start of the project, and then fine-tuned those pre-trained models using a small amount of prospectively collected label data. So this graph kind of compares what happens if you use this self-supervised pre-training step with supervised fine-tuning on just a small amount of data. That's the graph in blue, compared to a standard sort of fully supervised training using only the labeled portion of the data with varying percentages. And what we can see is that if you do the pre-training, then even with just a small amount of prospective label data, you can already get pretty good performance. So we say even at, for example, just 5% of the training data, of the label training data, we're already getting close to 90% AUC. Whereas a model that's trained from scratch would suffer significantly because of the lack of label data. So this is interesting in the case where we're faced with data scarcity. The other kind of interesting thing is that even with the pre-training, we actually improve the ability of models to focus on features that are relevant. So these are gradient saliency maps. The left on the right-hand side and the left column are we're showing two examples. The first row is the ultrasound image with the ground truth label. The second row are saliency maps extracted from just a standard supervised model where you can see that the model is able to detect with pathology, but it's also identifying other structures on the sides that actually aren't part of the positive class. And whereas if you look at the last row, the saliency maps are much cleaner, and the areas that are highlighted are mostly within the pathology region. So this pre-training not only allows us to work with less data, but also improves the robustness of the models for localization. In addition to unlabeled data, it's also possible for video-based modalities to work with partial or weakly labeled data, right? So a few slides ago, we compared the time required to frame annotate videos versus just a more simple video level annotation, and that time difference is significant. So what we worked on here was to implement or develop semi-supervised learning methods where we can combine a small amount of frame labels with a large amount of much more efficient video labels. And this is also kind of taking a book out of the contrastive learning page. Here we are applying a teacher-student training approach where we augment the teacher and the student differently. And the teacher serves to produce pseudo labels during the course of training, and pseudo frame labels, I should say. And these pseudo frame labels are then used to guide the student. So we kind of bootstrap the performance of the student based on the gradual improvement of the teacher. And we can then supervise this also with just a weak video level label to improve the quality of this teacher-student mutual learning step. And there's some additional technical details that we implemented to improve both the way that the teacher and student update from one another adaptively, as well as ways to ensure that the pseudo labels are of high quality and that we're minimizing the detection of false pseudo labels. And these cleanup steps improve the ability of this semi-supervised learning approach to work. And so we use this method on a data set of 300 something frame label data, but a much larger amount of weak video labeled data. And what we showed is that compared to a baseline model, this is a YOLO detection model in this case, trained only on frame label data, using this weak data set, in addition to the frame data set, allows for not only an improvement in performance, but also less variability in repeated training runs. So it also leads to stabilization of model performance over repeat experiments. And just some visual examples where these are just cases where the baseline detector would detect either false negatives or false positives, and those are corrected when we incorporate the semi-supervised approach using the large amount of weakly labeled data. So the takeaway, our learning from this is that when we're faced with labeled data scarcity, methods such as self-supervised and semi-supervised approaches, leveraging unlabeled or weakly labeled data, can both save a significant amount of annotation time and also improve model robustness. And one other challenge related to model development is that medical data sets are often highly imbalanced. So this image shows the distribution of different pathologies in terms of in the data set. So we collected this data set of 15,000 something videos, and we looked at the relative prevalences of each of the pathologies. And what we can see is that the data set is always highly skewed toward negatives. And this is Not surprising, I think this is consistent for a lot of different medical data sets. The positive class, and in particular, the more severe positive class, is usually a small percentage of the overall data set. And so we end up with something that's very far from an ideal sort of 50-50 positive negative balance. And when we consider the severe subclass, that skew is even more exacerbated. And we know that models often exhibit bias toward more dominant groups and lower accuracy in less prevalent groups. And this is a problem because the severe cases are often the cases that are most clinically important that we want to make sure we get right. But they're also the most rare cases where the model sees the fewest training examples and has the highest variability in performance. And so the question is, can we somehow increase the representation of low prevalence data so that we have more balance and we can improve model reliability in those underrepresented subgroups? So here I'm showing kind of prevalence in the context of feature distributions, but you can also think about it in other dimensions such as demographics, for example, where you want to make sure that the models aren't biased toward more dominant demographics and still perform well in less represented patient populations. One approach that we've taken to try to boost up the performance of low prevalence subgroups is through synthesis. So here the goal is to synthesize realistic images and kind of use them to augment real images. We can combine the synthesized and real data to train downstream tasks and using a richer combined data set. So what we did here was develop a method that actually synthesizes images using latent diffusion. But rather than trying to kind of use diffusion models to create medical images fully from scratch, we applied a lot of strong conditioning elements. The most important one is that we provide as an input into the diffusion model pairs of healthy and pathological images. So we would take a healthy image, and then we would sample from a bank of pathological images an appropriate image. Then we would kind of take the pathological regions and create a latent structural representation. This is what we're calling the pathology, skeleton S. And the negative image combined with this structural representation of pathology, those are encoded and then sent as conditional inputs into the diffusion model. And then the diffusion model creates a synthetic image that's based on that paired healthy image and those pathological representations. So why go through all of this? The reason is, it's risky, I think, in medical images sometimes to synthesize medical images without conditioning. If there's cases where the images are unrealistic or have some sort of bias, it becomes harder to control. So by conditioning these on real images, we kind of constrain the ability of the model to hallucinate or to synthesize unrealistic images and ensure more realism here. Because of time, I won't go into all of the details, but how to actually sample negative and positive pairs is really important. In this case, we considered the locations of pathological regions relative to neighboring anatomical landmarks. So like given, for example, a pleural line, which is a standard anatomical structure in the healthy image, we look for pathologies within a certain probability distribution based on the spatial location to the pleural line. And this ensures that the pathology that's synthesized is in reasonable locations on the image, and we're not, synthesizing pathologies where they shouldn't be. And so here on the left-hand side, we just show some examples of real and synthesized images. And over time, with a lot of iteration, we were able to start to generate images that were visually realistic. And interestingly, what we also found is that if you combine the synthetic data and the real data, you actually can improve performance compared to training on real data alone. And most notably, that improvement is the highest in the least prevalent groups. So the table on the lower right there shows the data set broken up into different feature severities. We can see that the green one and the red one have the lowest percentage of positive cases, and those also happen to be the subgroups where the inclusion of synthetic images seem to be the most beneficial. So this would suggest to us that a method like this could improve performance of models, particularly in those areas where the real data are the most limited. So, just to summarize the takeaways, that enriching training data with synthetic images can improve model performance, especially in low prevalence subgroups. And then finally, we want to compare the AI and human expert performance, right? So here, human experts were measured based on the agreement between pairs of annotators. We wanted to look at how well annotators agree with each other, and then compare that to how well the AI agrees with annotators. And what we were able to find over time is that the AI is able to actually outperform individual human experts in terms of detection and diagnosis. So that AI models can exceed the inter-rater agreement of individual experts in detecting and classifying lung pathologies. That's not to say that the AI is better than humans together, but I think AI compared to individual ratings appear to be more reliable across this set of pathological features that we've been working with. And I realized that we're sort of running on the hour, so I'm going to go through the last part a little quickly. But I think this part is really important. It's how to actually demonstrate real-world efficacy. One element is understanding how the clinician and AI will actually interface with each other. And medical AI models rarely work in isolation. Certainly today, they usually work alongside the clinician. And so it's really important to understand how the AI fits into the clinical workflow and how that facilitates clinician interaction. And the FDA also has guidelines as part of their good machine learning practice guidelines on this topic. Here I'm just pulling a figure from a paper from McNamara et al. In 2024, and they nicely summarize the different types of CAD devices. So CAD stands for computer-aided detection. It's a very common type of AI-based device according to FDA classifications. And there's actually 5 types of these computer-aided devices, shown here: CAD-T, E, X, EX, and A. And what we've learned is that it's very important early on to understand which type of device we're working with and where that AI is intended to interface in the existing clinical workflow. So what we can see is that CAD-T, for example, is a triage device, and that's designed to flag cases. It could be to prioritize how cases are reviewed, the order in which reviewed, and those devices would interact with the clinical workflow earlier on in the process. Comparatively, for example, a CAD-E or CAD-X device that's focused more on detection and diagnosis, that's where our work has largely centered. And the goal of those devices are very different from the triage device. And then there's a 5th category called CAD-A, which is fully autonomous. So understanding which type of classification the model will fall into is important because it changes how data should be collected, what the performance metrics are, and then what the study design and what regulatory bodies look for when establishing clinical evidence through validation studies. And it becomes pretty difficult to change from one class of device to another halfway through a development project, because a lot of these elements would then have to be changed or perhaps even redone to some extent. And just also relevant is understanding or designing the type of prediction output and how models can be explainable. One thing we've learned is that doctors care a lot that they can at least understand how the model output is, where it's coming from. And so, for example, in our case, we've learned that being able to show where the model thinks the pathology improves confidence compared to just a classification without any other explainable output. So again, this paper summarized among all current FDA-cleared devices the different types of predictions, whether it's a binary prediction, a score, or a categorical prediction, and also kind of summarized the types of outputs from these devices. We can see that a lot of devices don't have additional outputs, but more and more we see devices that either provide some kind of localization or other quantitative information that can provide some insight into what the model is looking at. Another really important step is after the development phase is over, again, how to clinically validate these AI devices. And so once again, the FDA has guidelines for setting up clinical studies to evaluate AI devices. And there's two kind of common types of studies. One is what's called a standalone performance study. So here the goal is simply to demonstrate the performance of the model on its own terms. So the objective of a standalone performance study is to assess agreement with ground truth established by clinical experts. So in our case, we set up standalone performance studies involving multiple readers and videos, and the agreement is measured on a per video and per frame basis. So you basically run the AI on a set of images or videos, and then you have a panel of experts review and read those same set of videos, derive a ground truth vote to establish a label for each video or image, and then calculate metrics such as video or image level sensitivity, specificity, AUC, and so on. So this is more similar to how a lot of the development work is set up, where and the metrics are also common, technical metrics. But the other study design is called a clinical performance assessment study, and that takes the validation one step further. So here the objective changes. Rather than simply demonstrating the raw performance of the model or the algorithm, the goal here is to understand how the clinician using the AI is impacted by the AI. So the study actually compares performance of human experts alone versus human experts in combination with AI. So it's a human with AI versus human without AI. So you can see that that's a very different flavor of study, and in many ways more reflective of how the AI was actually used in the clinical setting. And rather than metrics on a video or image label, the true ground truth here is a reference standard on the patient level. So basically the way these studies are designed are we have a data set that we provide to human experts who provide diagnostic outputs on their own. And then there's often a washout period to avoid biases. And then experts are then provided with AI results in combination with the images, and then provide diagnostic outputs. And then we compare which one is better, which one agrees better with the true reference standard or the true patient disease state. And so again, the goal here is to mimic the actual clinical workflow as closely as possible. And these study designs are important and often required when submitting evidence to FDA and other regulatory bodies, where the standalone performance of the model is part of the picture, but alone not sufficient to gain regulatory approval. And just on the topic of regulatory approval in the US, at least, the FDA recognizes three main pathways that kind of go from the lowest level of stringency to the highest level of stringency, depending on the risk incurred by the device. So the third category, the most stringent, those are devices that really can risk human life. So they could be, for example, a cardiac pacemaker that uses AI inside of it, that has a direct impact on life support and potential high injury risk, whereas diagnostic devices, such as the ones we're working with, may fall closer into the first two categories. And the figure on the right-hand side is a nice one that summarizes all of the different types of devices and approvals and their clinical areas and the approval pathways that they underwent. So again, similar to the couple slides ago, understanding which classification of CAD device and where in the workflow of the AI falls, it's also important to understand which regulatory pathway will end up facing and having to navigate early on, because it greatly changes a lot of the upfront effort and how data are collected and how studies are set up. Okay, I think we're close to time, so I'm going to wrap up quickly. The final important piece is real-world testing. And this can include testing even after deployment. So data set shift is a major cause of AI performance decrease post-deployment. And this occurs when there's changes in any number of different parameters after a model has been released into the world. So this could be changes in technology, for example, the imaging hardware changed, or the populations and settings where the disease changed, or the way the AI is used, the clinical practice setting changed. It can also be behavioral. So over time, medicine also changes, and maybe when the AI was first developed and cleared, how clinicians work with the AI is different than what it eventually becomes over time. And so if models aren't updated, then they may drift away, and you see that in terms of a decrease in efficacy. And similar to other points from before, regulatory bodies have come up with guidelines for how to identify and mitigate data drift. And common best practices include setting up processes to monitor the performance of the AI after deployment, and even potentially update the AIs either incrementally or in a continuous manner. And so this is kind of the final piece of the puzzle is even after freezing the AI, validating it, and getting regulatory approval, it's still important to ensure that the AI continues to work well after it's deployed out into the world. And so in our case, what we are trying to do is to set up post-market surveillance studies across different clinical sites, and also post-market clinical studies, so that we can carefully assess the impact of these models in the actual clinical workflow, and look at not only technical performance metrics, but also impact on clinically relevant parameters and metrics, such as time to treatment, diagnostic outcomes, clinical confidence, healthcare cost, and so on. So just to wrap things up, to conclude, there's a lot of challenges in translating AI to the clinical practice, and here we tried to cover a few of them related to data, annotation, model design, real-world efficacy, and some of the learnings are that data quality is often more important than quantity. We need to ensure the data are diverse and representative. Annotation standardization is a very important step. In terms of models, we want, if possible, leverage technical methods where we're faced with label data scarcity, explainable outputs, improve clinical confidence, look at underrepresented groups very carefully to avoid bias. And then in terms of real-world efficacy, assess the impact on the clinician-AI team instead of the AI alone. And then finally, ensure regulatory alignment early on, and establish processes for testing, real-world testing, even after deployment. There's one final component, I think, that's crucial to success, and that's a multidisciplinary team. So wanted to end by kind of acknowledging the entire group and highlighting the diversity of expertise that's needed to make AI a success. So of course, the technical work, AI development, and research are crucial. In addition to that, though, it's very important to have software user experience design, software engineering to ensure the integration is seamless, to have, of course, clinical science, program management, clinical affairs, biostatistics, regulatory experts, quality experts, clinical domain experts, clinical operations team, legal counsel to understand the ethical elements of AI, and then finally, clinicians themselves, so clinical partners who both help with the data collection and annotation and provide their clinical expertise based on their experience in the field. So some references here and end here. So thanks, and happy to take any questions with the time left. Thanks.
Thank you. Very nice talk. Thank you, Alvin. We have questions in the Q&A box. The first question, could you share your slides and references? So we will contact the speaker for the permission afterwards. Second question from Chang, how does data augmentation help the data diversity problem, if at all? Are there any guidelines of how we should use data augmentation?
Yeah, thanks. It's a good question. I think data augmentations is really critical. Kind of one principle that we've sort of adopted is augment to the most extent possible that still allows a reliable model training. So if you're comparing two models that have similar performance and one was trained with very strong augmentation, I think that model would be probably more reliable than a model trained with less augmentation. So when it's hard to collect data that covers every different possible variety, I think augmentation can help. I do think that there's limitations to relying only on augmentation. Um, I think it depends on the type of augmentation. For example, classical ones, rotation, translation, gain, and injecting noise and things like that. Those can improve certain elements of robustness, but they won't replace missing types of data altogether. So if you have an entire subgroup of disease that's not represented, it becomes much harder to create that through augmentation alone. The work on synthetic image generation, that was kind of one step that we took to try to improve our augmentation process, where we extend augmentation beyond the more traditional methods to synthesize more realistic pathology in underrepresented areas. So I think that type of work is a potential direction where we can leverage what we call quote unquote advanced augmentation methods and supplement that with the real data set.
Thank you. Next question, how the two groups of annotators are defined?
Yeah, so good question. So the two groups of annotators, I guess there's different ways to define it. The very first way is to make sure that annotators that are used in the training and development of the model are different than the annotators used in the clinical validation step. That's actually one important finding that we learned over the course of a lot of this work is I talked about separation of training and testing. That includes not only the data, but who annotated the data. And so the FDA and other regulatory bodies care about the fact that the model isn't biased to a specific group of annotators. And so when clinically validating the model, it's best practice to actually bring in a different group of annotators and demonstrating that model performance still holds up there. In terms of development internally, it's typically random. So we try to randomize the distribution of annotators that are assigned to each video so that they sort of average out and we have a broad and non-biased representation of the individuals in the development data set.
Thank you. Next question, did you keep annotators' performance blind or did you give feedback to specific annotators if they deviate from the group significantly?
Yeah, another good question. So it's tricky. We try to make it as blind as possible. I think these are experts that we're bringing in. They've been practicing for years, and before they join the project, we look at qualifications and things like that. Once they start annotating, we try to have as little influence as possible because it's always possible that then we inject our own biases into their annotation thought process. So we typically only try to provide more guidance individually when there's a kind of an obvious outlier, when we see that one annotator is behaving noticeably different than the others, then we might talk to them to at least understand their rationale. And sometimes we learn one thing or other that causes that difference, and then make a decision whether or not it needs to be addressed, or if that's just part of the natural inter-rater variability that's to be expected. Having said that, like as I showed in one of the slides, that upfront work of defining the annotation definitions and then making sure that all of the annotators understand the definitions in a consistent manner before the bulk of the annotation work progresses is important. So you want to make sure you get everyone on the same page, and then after that, try to allow them to use their expertise. And in our case, we try to influence them as as little as possible. And
Thanks.
in terms of, sorry, just to
No,
in
sure.
terms of the validation, actually, there's pretty strict criteria about not affecting annotators in the validation step. And so in those cases, ideally, the annotators truly are working independently and using their own clinical expertise.
Thank you. Is the annotated consistency study available to read as paper?
Yeah, this is a paper that was published recently, so happy to, I think it's in one of the references, and happy to share the paper. Sorry, Khan, I don't know if there's connection issues again. I'm not hearing any audio.
Yeah, yeah, so maybe if we are able to put the videos on the YouTube, you can let me know a list of references that you would like to add to the video description. Okay, oh, cool. It's
Yeah,
back.
absolutely. So happy to share any of these. Some of this is our work. A lot of it is other papers from others, and also there's I tried to list down some of the FDA guidelines that we found very helpful. So happy to share these, however makes is easiest for everyone.
Thank you. Next question, what would be the optimum number of annotators? How did you decide on it?
That's a hard question. I think in our clinical validation studies, the typical number that we've worked with, and also we've seen other approved devices use in the validation step, is anywhere from 3 to 7 annotators. So what you see is that individual annotators have a relatively high disagreement rate. When you then take the majority of 3 annotators, that agreement improves. When you go to 5 annotators, it improves a little bit more. If you go to 7, it improves a little bit more. Eventually, it starts to plateau. So there is some diminishing return in going beyond that number. So if I had to just throw a ballpark out there, 3 to 7 annotators to annotate each video is sort of the established range. Similarly for development, I think most of our data were annotated by 3 annotators. There were some cases that were sort of a challenging subgroup where we involved 5 annotators. The more the better, but of course that comes with time and cost. So there's a trade-off. But yeah, anywhere from 3 to 7 is I think that the most common number that I've seen in cleared medical devices.
I continue the question about annotated consistency. Can AI also approximate the variance of the experts? More disagreement between experts may imply a hard case or poor data quality. Would love to know your experience as our lab does not have access to multiple annotators. Thanks.
Yeah, it's a good question. So I actually think that this is one aspect that is an opportunity for us to explore more. So we've taken the multiple annotations and basically aggregated them into a single consensus vote, majority vote, and we're not fully accounting for the variance in the annotations. I think the variance in the annotations itself is a very rich information, and you can think of different ways to leverage that. For example, there's work that tries to learn the variance itself. So if you have let's say you have an image that's annotated by 5 annotators, they all agree with each other. That's probably an easy, obvious case, right? And so you can actually provide the variance or even the individual ratings to the model as additional supervision. And then, in contrast, a video with high disagreement where one annotator says negative, another says severe, a third says in the middle, that's going to be a challenging case. And the model can be told that that's a challenging case if we provide variance information. So I think there's a lot of really interesting research in that direction. That's could potentially lead to even more robust models. The other aspect that I don't think we fully explored, but is also relevant, is the model itself may, as an output not only a prediction, but also either a confidence interval or or a standard deviation around the prediction, and those standard deviations could be learned in the training process if you have access to multiple labels.
Thank you. Next question about synthetic data. Do you think that synthetic data generation will be necessary to improve medical image models? In case of yes, does the synthetic data generation have any major defects that affects significantly the AI model's performance?
Yeah, I think synthetic data is quite new. I'm personally not aware of a cleared product trained with synthetic data, although maybe it's starting to happen in the most recent medical devices. It would be hard to say whether it's absolutely required. I think in our case, we have some evidence to show that it helps in kind of fill in the gaps where the real data is more limited than we want. I would say nothing replaces real data, and there are risks in using synthetic data. It is possible that the synthetic data has artifacts. We know about hallucinations in generative AI, for example. And so those are risks that need to be monitored. There's some interesting work actually where synthetic data are then provided to human annotators to review. And so then you can still rely on a human in the loop to do a quality check of the data. And in fact, that feedback then can be provided to the generative model to improve its own performance. So one can kind of design a feedback loop in that way to improve quality. So I don't know that it's necessary. I think the vast majority, if not all, currently cleared devices are based on real data, but it's certainly an opportunity um, to improve model robustness where real data is lacking.
Thank you. Next question, does continuous training of AI model violate their validity in terms of FDA approvals? I mean, can we change the model once this is approved?
Yeah, that's also a good question. So this is also very recent. I think just this past year, the FDA came out with guidelines. This is one of the references here. It's this predetermined change control plan. So they've established some policy around how models can be updated post-deployment. Basically, the general idea is that during the submission itself, in addition to submitting the evidence related to the current model, you also submit a plan for how the model will be monitored after deployment and how the model would be updated. So the FDA actually reviews that plan as part of that initial approval process. And if it approves this predetermined change control plan, then after deployment, you can follow the proposed plan to update the model. And so that can be at whatever increment makes the most sense for the model. An example could be, for example, we submit an initial submission to the FDA, and then we propose that we're going to add more data over time. So maybe some point in the future, we extend the data set. We double the data set, and we propose a way to add that data set into the existing data and then retrain the model. And then we propose a way to revalidate the model, either using the same validation data or perhaps having to collect a different validation data. So that's proposed upfront, and then the FDA reviews it. And if the FDA clears that plan, then it can be implemented. So that's kind of the current regulatory framework for adapting models afterwards. Real-time adaptation, I think, is very new, and it's mentioned in this predetermined change control policy framework, but it's very new, and I would say it's something that the FDA is also trying to understand how to properly regulate.
Thank you. We are we are out of time, and there are over 20 questions in the Q&A box. So Alvin, would you like to pick a couple of questions from them to answer, and then we will end this. Yeah.
Sure. I'm actually gonna, okay, let me look through this. Multi-data integration. Yeah, also important. I think right now this is another unexplored area for us. Our
So
work.
Yeah,
Oh,
sorry to interrupt. Could you read the question?
oh, sorry. Yes. So
Yeah,
what
sure.
the question is, have you explored multi-data integration? Do you think this would enhance point of care diagnosis? I think multi-data integration is kind of the next frontier in a way. I know there's a lot of work already being done in that area. We're starting with one modality at a time, focusing on ultrasound. I think the key point in our case is that we're not making the clinical diagnosis directly, right? We're detecting features and pathologies and providing that information to the clinician to make a diagnosis. The actual diagnosis that done by the clinician takes into account a lot of different data sources, not just the image. And so if we want to develop AI that actually produces a final diagnosis, I think that almost requires multi-data integration. And so I would say that as you move from feature level assessment and detection, which is what we've kind of been focusing on up to this point, into a more holistic clinical diagnosis for a patient. Multi-data integration is, I would say it's required if one wants to actually develop AI for that second task. Um,
Thanks.
Let me just keep scrolling down. So how do we ensure that synthetic images cover both spatial expression of disease as well as severity? What about uh, commodities, particularly since the premise is limited data? So spatial expression actually is the reason that we set up that sampling step that I kind of went over. So we wanted to make sure that the pathology is properly distributed. So given a negative image that doesn't contain pathology, if we want to realistically add pathology, the relative location of the pathology becomes important, right? So let's just, for example, if we're trying to add pathology to the heart, often in an image, there's other structures outside of the heart. If you add a cardiac pathology into the heart, or that's supposed to be in the heart somewhere else, it becomes totally unrealistic. And so that's one of the risks, I think, of generative images is that we have to ensure that the generation is realistic. And so the approach we took was to carefully sample the relative spatial positions of the pathology compared to landmark structures in the image and make sure they fall within the expected spatial distribution based on our real data. And so that sampling step is one of the guardrails that we implemented in this work to try to ensure realism.
Thank you.
Okay. I can keep going. So have you come across significant additional training need post-deployment? If yes, have you come across high computing GPU needs, which is an obvious challenge in the clinical setting? Would lightweight models be an option here? Are there other suggestions? Yeah, so lightweight models are particularly important in our case because a lot of the models that we're working on in the ultrasound domain actually have to be deployed on lightweight hardware. Of course, some AI deployed on the cloud, and then you have access to a wider array of possible computing infrastructure. But for AI that needs to be deployed on the imaging device itself, their computational constraints start to be really important. For example, some of the ultrasound devices they run on phones and tablets. So the computer is actually the phone or the tablet, and you connect the imaging equipment to it. And so there, of course, the model needs to be lightweight enough to be able to run, on an iPhone, for example. And ultrasound is video-based, and so it also needs to be run in real time without lag. And so we all, in our work in particular, we typically work with lightweight models. A lot of these are out of the box. YOLO, for example, is a super popular detection model that we've had success with. And part of the reason is that because it runs quickly. So I would say it depends on where the model runs. If it's cloud, if it's on a device directly, that has a large impact on computing requirements.
Thank you. Maybe we just got time for one last question, then we will end this talk.
Sure. Let me see if inter inter-device variability was a question. So that's also important. we have seen that training a model on one device and then testing it on a different device leads to performance drifts. So that's one of the forms of data shift we're that can impact model performance after deployment. I would say that it's pretty important to cover at least a representative array of device types and vendors even in the development process. Augmentation could be able to help. So if one could understand the sources of image differences from one type of device to another, whether it's resolution, noise, underlying physics of how the images are generated, then some of that could be simulated. But short of having realistic simulation of that variability, it is important to ensure that different devices or any devices that the model is intended to run on is represented in the training as well as the validation data set.
Yeah, thank you. Thank you so much for the wonderful talk, and thank you for all the audience. And so that's the end of today's talk. And if we got permission from the speaker, we will post this recorded video to our YouTube channel. Thank you.
Great. Thank you, Khan. Thanks, everybody. Bye.
Thank you. Have a nice day.